Thank you for the introduction. Almost lunchtime, so I try to be quick. New traits related to feed efficiency, topic of my presentation, because this is really, the core of what we are doing within Feed-a-Gene. So briefly, a few words about Feed-a-Gene, but you heard the introduction earlier this morning. The main core of my presentation will be to talk about new traits related to feed efficiency, and I will provide you with a few of examples of working activities we are undertaking within the project, and one slide on future applications. So this is the one you have seen before. What is the objective of Feed-a-Gene? We consider the feed on the one hand and the animal on the other hand. So we bring together two fields of research, two fields of interaction within, let's say, the animal production column. And we try to bring these two worlds together by also considering, let's say, models which tell us more about how to include new traits into practice, and to understand if we turn the button at the left side, how does the performance of animals or the efficiency of animals respond to that. Too much to say within 20 minutes. But basically, and that's already stressed by Valerie early this morning, it is about using and understanding variation in production performance of different animal species. And in particular, we focus on pigs, on poultry, and on rabbits. On the one hand, observing the variation and trying to understand variation. Why do some animals respond differently compared to others? And at the right side, to use this variation and this knowledge to predict what an animal will do, how it will respond to a particular supply of nutrients in the diet towards end products, let's say body weight gain or amount of meat produced. And in the end, by understanding this, we want to use this by developing precision feeding systems. So better match the supply of dietary nutrients with what the animal is requiring for maintenance and production. And of course, we consider in this also the breeding sector, because in the end, we want to come up with new traits which can be taken into account by the breeding sector to make finally the outcome of feeding animals more predictable. So what's the trait? I will be very brief. There are many differentiations of what is a trait, and sometimes we talk about a phenotype, which is basically the response of a trait, but in any way, it's something which can measure on the animal. And even more important, it's something which we think we can change. So it's the response of an animal. It can be body weight gain, it can be the color of the eyes of an animal, or it can be something related to the fertility of an animal. If you want to understand these traits, you have to going deeper inside the animal and measure more of the different subsystems, organs, tissue, how they function, and what factors do interfere with the functions of these organs and tissues. So you have to go more deeply into, let's say, the DNA and the RNA of the animal, and try to see what kind of changes you observe there and how to translate that and try to understand that going back to the trait we are normally measure and have a more practical feature. So that means we also have to deal with the metabolites, and we'll come back on that, which are produced at different locations within an animal. So basically here, this is where the worlds meet. A phenotype is the result of a genotype, so that's the breeding sector which is involved in our project, and environmental factors, and one very important one, at least from my perspective, is nutrition. So learning more about this is what we are aiming for. And here, because that was the title of my presentation, feed efficiency. Well, to recall, what is feed efficiency? Most often we express it as the amount of feed required to obtain 1 kilogram of weight gain. However, you can, rather than kilo by kilo, you can also indicate that on an economic scale, or you can put the reverse, then we call it feed efficiency, but basically it's all the same, but you can express it in different units as indicated here. So feed conversion ratio has very important implication for practical animal production, because there is a straight relationship between the profits obtained in animal production and the average daily gain and the feed conversion ratio in growing animals. Here you see the trends in the Netherlands for pigs in terms of average daily gain and feed conversion ratio corrected for energy intake, and you see the gain is going up and the feed efficiency of the feed conversion ratio is going down, meaning that the animals become more efficient. On the one hand, that's related to the animal breeding sector, who does a good job. But secondly, this is also the result of matching better or dealing better with the environmental factors and providing the animal better feed. So here you see that expressed in a slightly different diagram. At the left side, it's the dietary supply of nutrients, which is related to ingredient composition, nutrient composition of the ingredients we're using in the diets. That relates also to the digestibility. We had some examples of in the previous presentation. We not only have to consider the gross presence of nutrients, but they should be digestible. And of course, the amount of feed ingested is determining the amount of nutrients consumed. That's the left side, that's the diet side. And the requirement side has also different components. We need nutrients for maintenance purposes, for just keeping the animal alive. But we also need nutrients and energy for locomotion behavior, and of course for deposition in organs and tissues. And you can also translate that to deposition of chemical constituents, and protein, fat, and ash and water are the main components of growth. The traits we are especially considering, and I have a few examples in a minute, relate to behavior and welfare of animals, individual feed intake of animals, digestive efficiency, and metabolic efficiency. Here, an example of one of the activities we worked on at the site of INRA on the one hand and IRTA in Spain on the other hand, where feeding stations were developed, allowing us to measure feed intake of individual broilers, individual birds, which are group-housed, or group-housed rabbits. So far, in group-housed animals, it's very difficult to discriminate between what is ingested by one animal and what by another animal. But knowing more about variation, and we are able to do so already for a long time in pigs, helps us a lot to predict the growth responses of animals. So here, some prototypes were built, and right now experiments are going on to identify what is the size of the variation and what can we do with the variation, also in terms of, for example, breeding animals which are more feed efficient. Another trait relates to improving nutrient digestibility. We know that digestibility of nutrients is related on the one hand to the enzymatic digestion in the small intestine, but secondly, and in particular in pigs, also a significant part of the animal is derived in the hindgut as a result of microbial fermentation of dietary constituents which are not degraded and hydrolyzed enzymatically in the proximal part of the GI tract. So what we are now doing with the omics technologies we have available, we are able to characterize the microbiota in the gut to a much more detailed extent as compared to what we could in the past. And that opens up also possibilities to see whether or not we see associations between digestibility of nutrients on the one hand and the composition of the microflora on the other hand. For example, we are very well able to discriminate diets. If you feed diet with a different composition in terms of ingredients compared to another, this is reflected in a different profile of the microbiota. However, what we did here, and this is just the first step, is to compare low and high efficient pigs. So pigs showing a high feed conversion ratio or a low feed conversion ratio, and whether or not we can discriminate the intestinal microbiota, in this case the fecal microbiota, in terms of their composition. And what you can see here, so the pink ones are for the high efficient pigs and the black response curves are for the low feed efficient pigs. We are able to see differences between these two populations of pigs. That's of course very interesting, because now we can have a look at what is the nature of these differences and what is the potential functionality of these microbial species in overall nutrient digestion in the pigs. This is really something new. Another phenotype which we are considering, and that in relation to nitrogen efficiency, is the birth weight of piglets. Birth weight of piglets is something we can measure quite easily, but in practical pig production, we are not taking into account in feeding pigs in later life whether or not it was a piglet with a very low birth weight, below 1 kilogram, or the ones which are above 1.5 kilogram. But it's very likely that the requirements animals have differing in birth weight will continue to exist throughout life. So what we did here, some nitrogen balance work where we consider nitrogen allocation between the proportion retained and the proportion which is excreted via either feces or urine, using low and high birth weight piglets. And this was just the first trial. Right now we are running a second trial, and we try to see whether we could find differences between high and low birth weight piglets. Of course, in absolute sense, there was a very significant difference, because low birth weight piglets continue to have a lower growth throughout life, generally, and vice versa. So here in the first trial, we did not see a large difference between the low and high birth weight piglets in terms of nitrogen efficiency, the proportion which is retained. But in the next trial, we also take into account the genetic capacity of protein deposition as an additional factor to discriminate groups here. So this is something which is ongoing and which has to do with efficiency, especially efficiency in post-absorptive metabolism. In the same trials, we also took blood samples to see whether or not we could identify biomarkers for nitrogen efficiency. So the proportion of the nitrogen, the protein which is ingested and which is used for protein deposition in the body, could maybe be related to some biomarkers present in blood. And right now we have the availability over metabolomics tools, omics tools, which via mass spectrometry could analyze a large number of metabolites at the same time. And that's actually what we are doing. And well, as you can see here, we are very well able to discriminate with this technology when we feed different diets. Here you see either a protein adequate or protein restricted diet. Is not so difficult to discriminate, but we also can see that we are able to discriminate the high and the low birth weight piglets. And this is a trial done when the animals are about 50 kilograms. So it's quite far in time from the time point they are born. So we are digging in further here to see whether we can find biomarkers for feed efficiency and nutrient efficiency. This slide was also shown before because in the end we try to develop new precision feeding technologies which will help us to bring the science also into practice, because more concepts which are targeted on precision feeding, that will be, let's say, the future of nutrition of our farm animals. In some species we are quite far. In dairy cows, for example, there we feed at an individual level, but in pig and poultry nutrition we are quite far from that, and I think we can make some steps there. Because what we see here, there is variation in the response of animals toward feed intake, that's represented here. But it also means that the requirements animals have are different. So if you group house them, you have 10 pigs in a pen, there will be differences in body weight, there will be difference in responses that mean the nutrient requirements, and in this case expressed by what they need in terms of lysine, being the first limiting amino acid for protein deposition, will be different. So this is something we have to take into account in feeding our pigs when we apply precision feeding concepts. For that, what we need is predict what, on the one hand, the feed intake of the pig will be if we know the history of the pig in terms of feed consumption. And secondly, we need to know what will be the feed intake on the next days and what will be the growth performance of the pigs on the next day. So by making use of historical data of a particular animal, we can predict, or we hope we are able to predict, on the one hand, the feed intake, but on the other hand, also the nutrient requirements of these pigs. And if we match, or we provide the animal with a diet which is targeted towards optimizing both the intake and their requirements, we think we can make a step forward in terms of improving feed and nutrient efficiency. And that's the concept which is indicated in this diagram. So we want to better know the response of the animals towards ingestion of a particular diet with a certain nutrient composition. And here is the response. Quite often it's the body weight gain, or it's nutrient retention or nutrient efficiency, or we want to identify a marker for this response, and we want to use this information for better feed, in this case the pig, for the next day. And here are a number of parameters related to the animal and the environment which influence this cycle which we generally apply in keeping our animals in production systems. So we have a specific work package devoted by improving precision livestock feeding, in particular for pigs and to some extent also for poultry, where we develop hardware components and decision support system to support the cycle, which I was just illustrating on the previous slide. And so it's all about data collection, data clearing, using the DSS to predict the body weight gain on the next period, feed intake prediction, and based on that also determine how many nutrients an animal will require on the next day. And if you have different diets available, which you can mix in different proportions, you are able to determine the supply of nutrients on the next days. And that's actually what we are doing now. So, final slide. We are working on many different new traits which interfere with the feed efficiency, and the challenge is to make use of this information considering the biological variation which these traits show in relation to feed efficiency, and using them for new precision feeding concepts is one very important application of these results. And secondly, we are in close collaboration with our partners from the animal breeding sector to see can we identify new traits which could also be included in breeding programs and the strategies of the future. And then they could relate to feed intake, to nutrient digestion, but also, for example, to animal behavior, because there we see also quite large variation. And in the end, I didn't talk about that today, but you can also relate that to feed efficiency. So I think that concludes my presentation. Thank you.